Hello, my name is Dr. Nathan Kleczewski. I'm a field crop plant pathologist or field crop pathologist at the University of Illinois at Champaign. And today I'm going to be talking a little bit more about corn tar spot. We're going to, again, an overview as well as some updates. So this is a topic that a lot of people have interest in because it's a new disease and we continue to learn more about it. So I just want to share with you a little bit about what we are learning in terms of this disease, how it works, and potentially how to manage it. So first, this is a fungal disease caused by bylacra matus, at least that's what it's called currently. It's an obligate pathogen. That means it needs a living host to grow and reproduce. It can't be grown on media currently. No one's been able to figure out how to do that. And we also don't have very reliable ways to research and study this pathogen in a greenhouse type setting either. It would need a living host to grow and reproduce. And we just simply are having a hard time figuring out how to get good, reliable infections in the greenhouse. We don't know currently of any alternate hosts. That doesn't mean there aren't any, it's just we don't know of any. We do know that it can overwinter in and on corn residue as black structures called stromata. There might be other ways it can overwinter as well, but again, we're still learning a lot about this pathogen. We do know that disease is favored by moderate temperatures, typically mid 70s to the mid 80s, and high humidity. Okay, so usually we have canopy closure. So after our canopy is closed, typically, that V12 to V14 is when we might first start seeing symptoms of this disease occur. And then after canopy closure, really wet, cool weather in fields where you had the disease, that's usually the ones that are going to see it first in those situations. So what are we looking for? Signs of disease. So the sign means you're actually seeing the cause of the disease, or you're seeing the pathogen itself. If you go out in a field, and again, you will see these kind of different looking black structures called stromata. They can range in size from being kind of roundish, as you see in this one here and some of these here. A lot of them also have this elongated appearance to it because you think about the veins of corn, it's a monocot. Remember the veins are going to run parallel to one another. It's a lot easier for these structures to grow within those veins as opposed to crossing the veins. And so that's why as these stromata develop over time, you oftentimes will see them have these kind of like whiskers at the ends that stretch out. So they might have a kind of a rounder part in the middle, and then the stromata will stretch out. If you do get into a field and it's humid and you kind of time things right, you can actually see ascospores popping out of these structures. That's what you're seeing here. I've seen orangish to yellowish ones here, like I did in this field in Monmouth this year, but I've also seen white spores as well from this fungus and how that relates to some of the things that we see in the field. I'll talk about that in a little bit. And then sometimes you'll also see that some of those stromata will have little halos around them. That's kind of what we call a fisheye lesion, although sometimes it kind of can look like this as well. I think that can be caused by all sorts of different things. We're not really sure about it, but we do know that we don't need to see those to actually have yield losses in our corn. As of December, this is our current distribution of it. This is from our EDD Maps site. Initially, the infestations kind of started here in Illinois and over here in Indiana. And then they kind of stretched out into Wisconsin, up in Michigan, and now it's been spreading. It's probably more established in Ohio than what has been assessed. It's just a matter of looking for it and reporting it. So I would say that it's probably, throughout this region, it's safe to say. It's also found in parts of Canada this year. And then this very strange observation right here of this isolated field and county. And I think there's some speculation that was probably pathogen coming in on infested materials accidentally. It's not uncommon for that to happen. This is carried on residue. We've seen images of bales being sold, and those are going to be used potentially as bedding or feed for animals. You also have decorative corn and things like that where I've seen tar spot on it. at places like Lowe's or even mom and pop shops. So it can move around on those materials, and that's one way it's going to continue to move around the United States. It's likely going to become established throughout this entire region, up through the Dakotas at some point, potentially up into New York, and maybe even further into these areas here, maybe parts of Kentucky. But right here, this is where we're dealing with it most, up by the Great Lakes, especially Lake Michigan. Now, how far can it disperse? This is something I observed while walking my dog, and I just think it's kind of a neat little observation. So I was walking and I saw my neighbor had this decorative corn that they had planted. And again, we live we're not in the country, right? We're in kind of our own little neighborhood here. So there's not any corn anywhere else. And I just kind of walked over and looked at those plants, and sure enough, there was a tar spot stroma on them. So you could see that, and it was pretty light infection. So this had to be coming from a distal source and blowing onto these plants at some point. And this is such a small bunch of plants, that means that there's probably a pretty sizable shower coming from somewhere when these landed, because that's a very small target to hit. So what I did was we did these concentric surveys of all the fields nearby that site. So this is the location where we had those 9 corn plants. And then I mapped out all the nearby sites within about 1.5 miles. I measured the distance, the least distance that would have to, if a spore was originating from those fields, would have to blow in order to reach those plots, and whether or not the field had corn in it this year or soybean in it, as well as the previous crop. So those spores could originate from the residue potentially. If it's a soybean field and it has a lot of corn residue on it, and that corn had tar spot the previous year, it also could be coming from this year's crop of corn and then blowing from that onto the plant, right? All of the corn that we had very low levels of infection. And we were looking at roughly, if we were looking at some of the old data that were coming out on this disease, saying that it only moves about 75 meters or 200 and I don't know, is that 250, 270 feet, something like that. In Latin America, we are, at least on this observation, saying, well, it can move at least 560 to 1,249 meters, or 1,800 to 4,000 feet. That's about 3/4 mile. If those spores came from the plants, those fields that I assessed. Now we see a lot of top-down infections, and because the infections were so light in those nearby fields, I'd say it's probably more likely that those spores traveled a much further distance from fields that had more established infections. So if we assume that it's coming from those fields, this would be the minimum distance they could travel, which is greater than the older reported distance. And this can be for many different reasons. We do have a flatter topography here. We do have those big wind events. We had a few of them this year. We had the derecho. We had in Champaign, we had some pretty bad winds moving throughout the season as well. So that's what you got to think about here, is as we learn more about this, you know, there's things that were in the literature that may not pertain to us in the United States. Also, we're learning new things that maybe when they did research on this in the past, they weren't able to do simply due to lack of resources and technology. So bylacra, the fungus that causes tar spot on corn, also causes tar spot on other plants. Typically, these are, if we're looking at the way these are defined now, they're actually moving the bylacra genus just into monocots versus another genus, which is going to have the dicotyledonous species in them. Because these are obligate, the thought in the past has been that they're very host specific. Because we can't work with these very easily, and we can't say culture them and grow them on plates, it makes it very hard to do these kind of confirmatory studies where we're able to actually prove the host range. So a lot of the work we're doing is in the field, and then you actually have to look at the DNA of individual stromata to see what you have there and how similar or different it might be from something else, from a different host. So typically they were just calling these different species on different hosts. But they were calling, if you found Bylacra on a given host, it was a different species. That's how they were defined. A lot of times they were also using morphological features of the fungus to define as a different species. Unfortunately, a lot of fungi are very, they have a lot of plasticity, and their spore types can change depending on conditions and even the host types that are there. We've seen this with other pathogens too. So there's not a lot of molecular data and ecological data to support a lot of this, but this is what was previously reported. So these are some images of Bylacra I found just walking around this year, as well as some Bylacra that we obtained from some of the national collections. What you can see, again, you can get that fisheye lesion on these as well. Oftentimes these are going to be elongated following the veins. You can see them on all sorts of different plants and all sorts of different locations. And there's been, if you walk in a prairie, typically around forest edges or near ponds and little creeks and rivers, that's where you're going to find most of these. They really like it humid and kind of shaded. And that's probably the same thing with our corn tar spot as well. So I don't want you to pay too much attention to this, but what our lab is doing is we're looking at some of the similarities and differences of the different species of, or reported species of Bylacra. So what we're doing is we have samples from all sorts of different hosts, as well as corn and different locations of corn in the United States, as well as Mexico and Latin America. And so what this graph is doing, just think of this as a road. And when you're in one, you take one path, everything in that area is going to be more similar than in these other paths. And so our non-corn Bylacra will kind of have this salmon color on the inside. The corn will be gray. And then the location, you can see where the location is based on the color and also the text inside there. So the first thing to keep in mind, the majority of our non-corn isolates here are, oops, I didn't want to do that, are grouping out together. So here's our first, this is our first little node, our fork in the road. So you take a left, all of these are going to be more similar to one another than anything in this other part of the circle. That tells us that, yeah, there is some differences. The stuff on corn is more similar to one another than these ones on some of these other hosts. Now, it is important to note there is some, this is just one gene we're looking at. This is the ITS region. It's a conserved region of many fungi. We're also doing this with multiple genes, and so I don't have that data for you yet, but you do see intercalation of these corn ones within the non-corn, as well as non-corn within our corn. So that tells us that this is much more diverse than previously thought. What we then can do is we look at this here, this little node is telling us, again, that this is different from everything here. And then this node here is telling us that everything here is different from everything here. And that's interesting because this is all coming from corn. And it kind of gives us this idea that we might actually be looking at two different subpopulations or potentially species of tar spot or Bylacra matus in the United States. The one in this population tends to kind of group out more with the Mexican isolates, and the ones over here more with the Puerto Rican, Dominican Republic type isolates. That's pretty interesting. I don't know if that spore color relates to this, or if this has something to, or if this can relate to aggressiveness or symptom development or anything like that, how it interacts with the host. I don't know, but it is an interesting thing to note. And we do also see mixing. So you might have a sample here on one leaf where one isolate groups out here and another isolate on that same plant groups out over here. So we do tend to have mixed populations here in the United States, potentially or originating from at least two different populations. And we'll keep working on that and updating you on that. But one of the things to keep in mind is, and what should you take home from that is with the intercalation with the non-host and non-corn and corn species, that one host to one species concept is looking a little wavy gravy. And like I said, there might be some sort of alternate host out there that might aid in the dispersal of this particular pathogen. But right now, we're not seeing any evidence of that yet. But we will continue to do some more work and see what the potential alternate host might be. So if you do see tar spot nearby, you know, it's unlikely that that's causing or moving tar spot into your corn, especially if you haven't had corn tar spot in your region before. But if you have corn tar spot in your region and you start seeing it move, it's coming most likely from those cornfields, not from ryegrass or something like that nearby. But just keep in mind, we're still learning a lot about this. In terms of what we see with, you know, back in the day, we talked about fisheye lesions, and it's very important for fisheyes to develop. If we look at some of the survey data we took from 2018 and 2019, the overall fisheye lesion incidence, so the percent of a leaf that has fisheyes around them, are very low, typically less than 1% of a leaf with fisheyes in our samples. Yet we can get up to 35, 45% severity on a leaf without significant fisheyes, and that leaf will shut down. Think again about how those stomata form. They're like pimples. So as they expand, they're going to be putting pressure on the xylem and phloem elements. If there's enough of them on a leaf, that's going to make it really hard for that leaf to, well, think of it this way, some of the carbohydrates and nutrients, those are going to be going into the fungus to support the fungus, but also that squeezing and pushing activities of the fungus is going to make it hard for that plant to move nutrients from the leaf, and that leaf can rapidly shut down, especially if you have a situation where you might be in some sort of a stressful condition, like you get severe tar spot and then maybe you hit a dry period or something like that. Okay, so the need for fisheye lesions isn't necessary to see significant yield losses, but you want, if you do get significant infections of bylacra, especially earlier on in the season, and it continues to build over time, you can definitely see those severe yield losses. If we look at, again, the idea was, if you saw fisheye lesions, it meant there was a second pathogen. We've done some work on culturing this out, and there's been some work in Michigan also using a non-culture based technique to assess the fungi associated with fisheye lesions versus not. And there's another project we're doing that we're finishing up with. We'll have more data on this as well. But we did collect isolates from colleagues in Mexico that were working on tar spot. And so they sent us their quote unquote monogramphella isolates, as well as tissues and DNA from their plants. And then we also isolated out fusarium from samples in the United States. And then what you can see here, the ones in the United States are red, the ones in Mexico and this particular tree are blue. And then what you're looking at here, this is monogramphella up here, clearly different. Again, here's the path in the road. These are clearly related and distinct from everything over here. So everything in this group is different from monogramphella, even that in Mexico. So we're not seeing monogramphella even associated when we culture these out with those lesions. What's interesting too is when we start looking at these, the Mexico isolates are kind of distinct from those in the United States. These are just fusarium species that we see in these fields. These are Equisetae, and these are a couple of the other species complex. We have all sorts of fusarium out there. They like growing on dead, decaying tissue. You know, they like it wet and humid. And you think about fusarium head blight on wheat, you think about stalk rots and things like that. These are the same types of species that we see out there. So I think just because they're associated with those lesions doesn't mean that they're causing the lesions. That's something very important to keep in mind. And the other thing too to keep in mind is that, like I said, you don't need fish eyes to see significant losses. So bylacra needs to be the focus. And I say that for people that might be breeding. I think we should be rating the black spot development and not focusing on the necrosis. I think you're going to have much better and more reliable results if you're focusing on those stromata, even if this two pathogen system is required. If it let's say it is true that there is a second pathogen that's needed to produce those lesions. You still need the bylacra there to get those lesions to develop, right? So that is why we should, I think, be focusing on the stromata and not focusing on necrotic tissue when we're assessing hybrids, as well as performance of some of these fungicides. There's some work being done with my colleague, Tiffany Jamin, and her student, Sarah Lips, a master's student finishing up this year, doing a great job, this bang up job. One of her projects is looking at tar spot in what's these gem lines. So just think of these as really sources of resistance. They're publicly available. They come from all over the world. And then what Dr. Jamin did is she did some work on the overall set of like 500 different lines or whatever they have. And they found 50 that they decided were going to be kind of targeting a good, what should I say, most likely to be highly dissimilar and diverse from everything else in that data set. And so Sarah, yes, she's looked at several hundred of these in a large screen, but then she's been focusing on these more diverse lines in her studies to see if we can see any differences in tar spot resistance. And then if we can see those differences in resistance, that's going to provide potential materials for introgressing into hybrids. It can help us understand how resistance works, host plant interactions, so on and so forth. So she has 4 locations in 2 years. One of these locations was, we call it a V3 site. So this is planted very late. And so those plants were at the vegetative stage next to a field that was significantly infected with tar spot. They had tar spot then naturally infect those plants and infect them very early on in development. And so what she saw here was these numbers here, 0.83, 0.72, and 0.9. These are correlations between the different data sets. And you're seeing in these 3 data sets, they're highly correlated. These are the sites in those 2 years where we were looking at mature plants. The one at V3 doesn't correlate very well with the other ones. So it's kind of interesting. There might be something ontogenetic going on, so something age-related, but it doesn't appear like when we look at these lines early that the resistance scores correlate with those resistance scores later on in their growth and development. But what she does see here is we look at the Audpc, that's the amount of disease over time, less disease on the left, more disease on the right. She has some of these colored here so that you can see where these kind of spill out. And what you're seeing is that the best 3 tend to show up here on having the lowest amount of disease, whereas the worst 3 kind of pop up here on the top. But we don't see that again with the vegetative ones, although there are a couple of those that still pop up, but there's not a good correlation there. But what this is telling us then is that we have some distinctly top performing lines that have resistance to tar spot, and then we have low performing lines that are very susceptible to tar spot. And so what that's going to do again is give us some information for what to potentially look at for breeding, how to study where, these genes and how they work, so on and so forth. Tools potentially to develop markers to make sure that we can get the resistance in there, although this is, I believe, a multi-gene type resistance. But that's, again, I'm not a breeder, so I don't want to get into that. That's something for Dr. Jamin to talk about. In terms of fungicide efficacy trials, we've done some on our own. We've also done some coordinated trials with colleagues at other states. So I'm going to provide you some of that information here. These were the products that were tested or that were selected for testing. I'm only going to present the data from this year because I think it's much cleaner than the data last year. Last year, disease didn't show up till very late in the season. And so the data sets are more indicative of a residual trial. I think this year we had much better luck, at least in Illinois, and that's what I'll focus on for our results. And I think our trial is a pretty good litmus test for a lot of these fungicides. So if you look at these, again, these are the trials this year, and these are the different sites. Again, if they have different letters, that means that the averages here are significantly different from anything below it. So an A is different from a C, a B is different from an A, and so on and so forth. A and B would be similar to an A and a B. You can see we have different levels of diseases at the different sites. I'm not going to look, I'd say don't pay attention to this average. I think we need to have like a % control so we can standardize these and have a better idea of how they perform across environments. So just look down. And what you're going to see is that yet all these products, or most of these products, reduce the amount of disease relative to the non-treated checks when you have significant disease. And that's what we saw at these 3 sites this year. And they're also fairly consistent, right? So some of these are going to pop out as being better than others. 2 products in our trials, and I'm going to show more data on this, that aren't doing so hot are these 2 here at the bottom. And again, I'd say that that's supported by these other sites as well. Okay, so these are the data. So they are what they are. Not going to argue with that. If we have significant yield differences, it's going to be, we're going to see the impact of these fungicides in terms of benefiting these plants. Now, I don't know much about these sites here in Michigan or Indiana in terms of other diseases that were present. So I don't know if this 216 to 237, for example, is just tar spot or there's other things there. I can tell you here that we had southern rust, pretty significant on this trial as well. And so the reasons or a couple of reasons that you might see, 1, such low yields, and 2, maybe larger yield differences here is also due to the effects of those fungicides on southern rust. Now in Monmouth, we ran a multiple company trial. This is one, again, we applied it right at R2 and the symptoms started to just develop in the mid canopy and a little southern rust started to show up too. Just that same trial I was reporting earlier. These are the products we use. So you're going to see some things in there that weren't in those other trials like Delara Complete, Affiance, Badge, Domark, Proline. I don't think Proline was in the other one, but there's some things that weren't in the other trials. These are the symptoms we saw. So you can see here a mix of tar spot and southern rust. Really good diseases. This is the non-treated control here, the 2 different ratings, 41 days after application and 49 days after application. And then within those bars, different letters indicate different, statistically different performance. I'm focusing on the 49-day after rating. What's important here to look at is look at this difference from 41 to 49 days. This is why getting that on at a good, at the appropriate timing is going to be important because it has this kind of exponential growth and it has a very steep inflection point. So once it takes a long time to get going potentially, but then once it takes off fast. So getting that fungicide on at the right time can really help you reduce that disease. And then you're seeing here with these products, most of these, except for Proline and a Tilt, are really doing knocking down disease relative to our non-treated checks. But the ones that are doing best here are highlighted by those red arrows. So we have a lot of good products out here, and some appearing to do a little better than others, at least in this trial where I'd say this is a near perfect, if not perfect application at R2. We also ran a vegetative corn trial. I'm not really going to talk too much about this one. In fact, I'm just going to flip through this one. It's showing you the same results, really. So we're applying these to late planted vegetative corn, and we're applying them at wetness. So we're really trying to put severe pressure on those plants, as well as look at some curative activity. And really what you're seeing here is our non-treated checks. Again, the Lucento and Tilt not doing as well as some of these other products. And so I think the take home here is that if you're going to be applying a single fungicide, if you're going in with those multiple mode of action fungicides, those are going to be your best bet. Going cheap and doing like a prothioconazole isn't going to likely give you much benefit at all, especially if you're applying it at VT and you get later disease. We also did a timing by program trial in Coal City. So these again are looking at different timings and products in combination, 2 pass programs. I don't want to spend too much time on this because we're still looking at this. Again, I use whiskers here instead of letters to kind of separate these out. So you have the different application times here where we put down the products, and then we have combinations. So let's say I'm a grower and I do my normal VT application, but then I'm scared, I'm worried about tar spot, so I do an R3, or I do an R3 and I'm worried, so I do an R5. When these 2-pass programs are made, are used, that second one, we always use tilt. And we did that because we wanted to kind of use the cheapest program possible. So we would go in with our quality product first, and we'd follow up with a tilt, you know, that cheap generic prothioconazole, so propiconazole at that later timing. And you can see there's differences in some of these within each timing. There's differences in the products, but really what you want to focus on here are these averages. So here's our non-treated check right here, VT, and then you're looking here at our R3, R5, and again, VT, R3, R3, R5. The reduction in disease severity really being driven down by that R3 application in these. And although you get some reduction in the R5, it's not as good as that R3, better than VT. And this is something we're seeing in several of our other trials as well. So when it comes to tar spot, you're going to have to scout your fields and pay attention to when the disease is showing up. And you're probably going to be putting that application on a little later. Again, we've looked at just R5 applications. This is one we did last year, non-treated checks here. And then we're looking at our tar spot and lodging. And I only put letters on ones that were significant. No significant differences in yield. Yes, we were able to reduce our amount of tar spot when we make those applications of fungicides at R5, but not a lot of disease in general. Not a lot of disease in general late in the season because it's obligate. You need a green plant to grow and reproduce. There's just not a lot of time for it to do that on those plants. And then the other thing here to keep in mind is, I mean, those plants are finishing up. So there's not much that it can do in terms of maybe yes, it can. If applying these did cause the plants to be greener longer, but that could potentially have a negative effect because you could allow the fungus to maybe, you know, spend more time and hang out longer too. I don't know, but it didn't impact our lodging and it didn't impact our yield. So again, a well-timed application is going to be helpful. We also did a fungicide tillage trials. These are larger plots, and then we're assessing a small area on the inside. I know I'm going long here. Basically, I'm just splitting this out in the main effects here in the fungicide and no fungicide. So what you're seeing in utilizing tillage in a field, we saw a 16% reduction and a 77% reduction with our fungicide application. This was a VT application here in Monmouth. And so these would be additive effects. So you could add the 77 and the 16 in these, and that would be the greatest effect, the greatest reduction in disease. I'm not saying you need to till your field or anything like that if you're no-till. This obviously is a disease that can move distances pretty great, but this potentially can reduce the onset of this disease as well as the local inoculum. The earlier the disease starts in a field, the more quickly you're going to hit that steep inflection point and see severe losses. So if you are practicing some sort of tillage, it can help. In this case, our tillage was taking down our residue from 80% to about 22%. So that's what you got to think about here is, you know, utilizing IPM and what that means. So here's what you got to do. Get out there and scout, look for the disease. Don't forget about all your more, you know, those other diseases that are really important that infect corn annually, you know, across different areas. You want to be selecting the least susceptible hybrid. There's no resistance out there, but most of our seed people now have ideas of which ones do worse and which ones do better. So if you're in an area where you have more tar spots, select something that's less susceptible, but don't go thinking that that's going to give you a ton of protection. Just remember, you're chopping up this disease into pieces and trying to minimize the amount of damage it does and minimize when it takes off. Minimizing irrigation. If you irrigate in parts in the state, you see that in some states irrigate more than others. If you see it present, that's going to be a problem, but You know, it's one of those things, do you put water on the plants because your corn needs water and it's rolling, or are you more concerned about disease? Those are the tricky types of decisions you have to make when you're working under the pivot. Tillage may provide some slight benefit, but don't go thinking it's going to save the day. Also realize you can get it blowing in from other fields, but when we're talking about onset, that's probably going to be a delayed onset relative to local onset of disease. And that's really where tillage can provide some benefit. And consider a well-timed fungicide if disease is present, especially if you haven't, you're kind of on the fence. You're going to be dialing in between that VT and R3 period, but probably a little later is going to be a little better in terms of protecting yourself from tar spot. But don't forget those other diseases. Here's my contact information. Sorry, I went a little late. I had too much information for everybody, but feel free to contact me. My number is 217-377-4406. Feel free to give me a call if you have any questions or you'd like some more information. I'm always willing to talk. Take care and have a great day.